First, we're going to look fertilization in plants. Before we're going to learn fertilization of the plants, we should revise the parts of the flowers. So let's draw a flower and name the parts of the flowers. Okay, this is the basic structure of the flowers, and we can name the first one. This is called stigma, and the long tube is called style, and the round shape is called ovary. So these three, stigma, style, and ovary, this represent the female side of the flower. So the female side is also called uh, carpel. And this one anther, and this is filament. This is the male side of the plant called stamen. It's a male. This one is a female one. Okay, then the base is called a receptacle, and this one is a sepal, and this are called petals. Right, the petals are the colorful plant part of the flower, and below that, the base, there is again a green color leaves-like structure, and this represent okay the sepal. Okay, then last thing, the ovary has this ovule, and ovule has ovum, and this anther has the male gametes, a pollen grain. Right, can you copy this one? Maybe make it smaller because there are, I think, 2 pages. I think it's enough to record everything.
Okay, sir.
Okay. Yeah. Okay. Now this pollen grain can move from anther to the stigma, right? So I represent this from this arrow. Now this can happen from the same flower, or it can happen from one flower to another flower. So if it is happening in the same flower, we call a self-pollination. So we can write it here, transfer of pollen grains from anther to stigma is called fertilization. Right, so that's important to remember. The fertilization is transfer pollen grain from anther to stigma is called a fertilizer. So the fertilization is a pollination. It's called pollination. So you can write this under the diagram. Transfer of pollen grains from anther to stigma is called pollination.
Okay, sir.
Now we look what is the next step after pollen grain deposit on a stigma. So let's start what is going to happen after that, right? You can see different pollen grains under electron microscope. So the typical pollen grains, they have a wall like this, right? And then there is a inner wall. So this inner wall is, so this outer wall is called exine. The inner wall is called intine. The outer wall is very hard, and it is made out of a special structure, a chemical structure called hard layer, made out of sporopollenin. Sporopollenin. And this is intine. Okay. Now it's important. This has inside there are 3 nuclei. This is a 1 and another 2 like this. So the first one, this one is called tube nucleus, and these 2 generative nuclei. This 2 is called generative nuclei. So this is the structure of a pollen grain. Can you also draw this one next to these parts of the flower?
Okay, sir.
Structure of pollen grain. So this is 1. This is 2.
Okay, sir.
Right now, if you look the first stage, there is pollen grain from anther deposit on stigma. Right? So this is the pollen grain. And this is the surface of anther, stigma, right? Surface of stigma. So pollen grain can absorb water and maybe some nutrient from the stigma. After that, there is a small tube growing out of the pollen grain, right? So this is called germination. That means the first step is germination. So pollen grain absorb water and nutrients from stigma. Then small tube grows out of pollen tube. So step one is the germination. So can you write it down? Step one is the germination.
Okay, sir.
Right, then we will see what's going to happen to this pollen tube. Right. Okay, in step 2, the tube is growing now. Downwards from the entering from the stigma, it's growing downward now. So this is the stigma. That's the style, right? Right now, if we look how this happening, this structure is called pollen tube. A pollen tube secrete enzymes because uh, what I'm coloring here is the tissue of the stigma. This is the tissue of the stigma. Now, if pollen tube wants to grow, pollen tube should digest the tissue, surrounding tissues like this, and they can get the space to grow, right? So this is possible. Pollen tube secrete enzyme and digest stigma and style tissues. Why they do this? Number one, to get the space. Number two, to get the nutrients. To get the space, to get the nutrients to grow. So if we look what is inside the pollen tube, now I just zoom this area and I draw it again. Okay, then I can see two generative nuclei, and the polar nuclei is in the front. The tube nuclei, right? Tube nuclei. This is the tube nucleus, and these two follow behind the tube nucleus. They are called again generative nuclei. So they are moving this direction. So pollen tube secrete enzymes and digest stigma and style tissues to get the space, to get the nutrient to grow. So this is the step 2. Now, can you also write this down, step 2?
Okay, sir.
Step 2. Now, next step, step 3. Right now, this pollen tube is growing towards the ovule, and then it reached to the end, right? So this represents the female side still, and this is the ovary, and this is the ovule. And inside the ovule, we can find the ovum, right? So this is the ovum. There are other different cells are also there. For example, here, two cells in the middle, two nuclei, 1, 2. And there are another 3 nuclei here, and another 2 nuclei around here. So these are 8, 1, 2, 3, 4, 5, 8 nuclei. So these nuclei, these two, is called polar nuclei. And this 3 nuclei, they are called antipodal. And these 2 nuclei are called synergid. So here 2, here 3 nuclei, and here 2 nuclei. And this one is the main nuclei ovary, ovum. Okay. Now the opening into this ovule is called micropyle. This opening is called micropyle. Okay. All right. So here is the pollen tube. When pollen tubes grow close to the microfile, we can still find these two generative nuclei. And when it comes to the microfile, the tube nuclei breaks down. So tube nuclei breaks down. So we don't see tube nuclei, it's a breakdown. So only we can see two generative nuclei. So this is the step 3.
Okay.